doing good. How are you? It's okay.
I'm good.
Um, so I've been trying to finish up with like the Biochem reviewing it. And I'm stuck on the pentose phosphate pathway, and like gluconeogenesis, but I got like the TCA,
Hmm.
like glycolysis and the like fermentation, like
Hmm.
that those stuff. But for some odd reason, my mind just can't click with the phosphate pentose, sorry, pentose phosphate pathway.
Okay, we could go over that. So,
Let me share my screen, I guess, because I had the document pull it up that we had last time. Um, yeah, share my higher screen. Um, so I guess like this is where in your document where you talk about the pentose phosphate pathway.
mhm.
And I guess like a question I would have is like, first of all, when would this happen as opposed to another pathway like gluconeogenesis?
Hmm.
Um, since it is anabolism, so it's building something up. It's like what I can understand so far.
Yeah, so I'm actually going to open up a whiteboard so I can draw this um, in a new way. So,
Okay.
do you see the whiteboard?
Yes, I can see the whiteboard.
Okay, so so its purpose is to generate reducing power by making NADPH and five carbon sugars like ribose 5 phosphate. Um, which could be used for like uh DNA, RNA, ATP, NAD, things like that. Um, and so although it's so typically catabolic processes would be oxidative and anabolic will be reductive, but pentose phosphate pathway, although it's anabolic, it's oxidative. So we could start off with a What molecule is this?
Um, there's a I, I want to say glucose, but I don't think so. Like G6P.
Yep. Glucose 6 phosphate. And you understand like how the ring form of glucose is predominant in solution, right?
Like the way that it's in solutions normally, or
So, for instance, I'll do this on the side.
oh, like that versus it being in a cyclic formation.
Hmm. So this is, yeah, the straight chain form, which is around 0.1% of, so 0.1% of glucose in our bodies are in this form. Now, I always say about hydroxyl groups, what do I usually say? If you remember, with
Like
them to think what? Nucleophile?
yeah, they're nucleophile.
Yep. And when you see carbonyl groups, think electrophiles. So what
Yeah.
happens here is this fifth hydroxyl will nucleophilically attack the carbonyl carbon here. And so that'll be like a ring closing, and that'll produce the ring form of glucose. And I should say that, so this fifth hydroxyl group, I'm going to color in orange. And that's the one that nucleophilically attacks that first carbon position, the carbonyl carbon. So that's the oxygen that's incorporated into the ring. And then, um, oh, and then I'll use maybe like this pink color to indicate that carbon, I mean, sorry, that oxygen, which then becomes the anomeric like oxygen in the hydroxyl group. That's what that one is. And then I can fill in the rest. So we have OH here, OH here, OH here, OH there, OH, 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 OH there. Now, you see, okay, so the anomeric carbon is this one and, that one. Now, when it forms the ring, this hydroxyl group could be uh, upwards, or you see how the sixth carbon here is upwards, right?
Mhm.
So
Yeah.
this carbon, this hydroxyl group, if it is also upwards, this is called the beta
Beta.
anomer, right?
Yeah.
The one on this one is opposite. It's down relative to the sixth carbon. So that's
Alpha.
the alpha, yep. And around 64% of the glucose in our bodies is in the beta anomer form, whereas around 36% is in the alpha anomer form. And that's because uh, the beta anomer has all, like if you drew it as a chair, all hydroxyl groups are equatorial, as opposed to the alpha, where the anomeric carbon in that case would be axial. So,
Mhm.
okay. So yeah, you're, oh, and then there could be like interchangeability between the anomers, and that's called mutarotation. Okay. So, okay. So now let's go back to pentose phosphate pathway. So we have our glucose 6 phosphate, and we do something to it. Um, let me see if I can bring something up here. The generalized biochem redox pathway I will bring up. See this?
Yeah.
So I haven't gone over this with you, have I?
No.
I did.
No.
Okay.
No.
Got it. So from left, so like, okay, we know from Gen Chem that oxidation is loss of electrons, reduction is gain of electrons, but that doesn't help us when we do like redox in like biochem or organic chem. So this is going to help you. So typically you're going to see for, oops, for oxidation, you'll see like one of the three, either, loss of hydrogens, gain of oxygens, or gain of bonds to oxygens. So if we start off here with this alkane over here with the CH2, CH2, and from left to right is oxidation. When we oxidize this, we remove the hydrogens, creating a double bond, and that's what we get next. And then after that, we gain an oxygen in the form of a hydroxyl group. So that could look like, so like this COH, H, and then this CH2, like that. Now,
Mm.
now if it's a tertiary alcohol, it cannot be oxidized any further. If it's a secondary alcohol, it could be oxidized um, into a ketone, but then it cannot be oxidized any further than that. If it's a primary alcohol, it could be oxidized into an aldehyde, which can then be oxidized into a carbox. Oh, and by the way, I should say, like, okay, from the alcohol where you have a single oxygen that we've added to the ketone or aldehyde, that's when we're adding bonds to the oxygen. All right. But yeah, if we
Mhm.
now from aldehyde to carboxylic acid, so we've already added bonds to the oxygen, so now we're going to add another oxygen, and that's why you get your carboxylic acid. And then you're going to add bonds to that new oxygen to get CO2. That's the final, you know, form that I can take if it's being oxidized. And Um, and also I wrote like PCC. So like they could also ask you about like a weak oxidizing agent like PCC would only be able to oxidize the primary alcohol into the aldehyde. But if you have a strong one like KMNO4, it can oxidize the primary alcohol like into a carboxylic acid. And there's actually um so this works for reducing agents as well. It actually shows up more often for reducing agents. So for instance, like this to this would be like NABH4, which is a weak reducing agent. But from this to this would be something like lithium aluminum hydride, which is a strong reducing agent. So takeaway, whoops, here is that so sodium borohydride can reduce aldehydes and ketones, but lithium aluminum hydride can reduce like these aldehydes and ketones and carboxylic acids, um amides, esters, carboxylic acid derivatives. So okay, now this generalized biochem like redox pathway we can use for this. So for this glucose 6 phosphate, right, we're going to oxidize it. And so we're going to use glucose 6 phosphate dehydrogenase. And remember your enzyme classes, that's an oxidoreductase, right?
Mm-hmm.
And NADP will be the one that gets reduced to the NADPH, which means that the glucose 6 phosphate must be oxidized. Now, what can we oxidize? Or, yeah, what can we oxidize here?
Um, like for the next molecule or
Yeah, like so what in glucose 6 phosphate can we oxidize?
because I guess like based on the list,
Mhm.
like it could be like a loss of a hydrogen somewhere.
So,
No, what
so
is it gain? Or am I
what functional group do we see in glucose 6 phosphate that we see here?
hydroxides.
Hydroxyl groups, right?
Yeah, hydroxyls.
And those could be oxidized into.
That could be oxidized into a I see this to a carboxyl.
So
I'm
what,
skipping.
what type of hydroxyls do we have in glucose 6 phosphate? Like, uh, do you have primary alcohol, secondary, tertiary?
Um,
I should say that these are H's. Yeah, I didn't draw them, but
so I guess like I don't know the difference between like a primary, tertiary, or like up there.
see what I'm drawing now?
Yes. So would the bottom one be a primary? Yeah. Okay. So at least this one, it looks like um it's a tertiary over here.
So I should also state that there's also an H there, and then here there's two H's.
Here. There's an,
Mhm.
then there's so it's connected to two different things in a hydrogen. Secondary.
Mhm. And what if you oxidize that, what does it turn into?
If you oxidize that, then it will turn into a ketone.
Yep. So here, this guy turns into a ketone, right?
Hmm.
And now there's a special name for what this is, which is a lactone, which is a because okay, what is this molecule? Like what type of molecule is that?
Um, I see both two oxygens here. Sorry, yeah, I'm blanking.
Sure.
Yeah,
So
yeah,
it's an ester,
yeah.
okay? So I guess over here, so like so an amide would look like something like that, right? And an ester would look like that, right?
I don't see it.
Oh, I'm drawing like all the way back down.
Oh, oh, I keep on trying drawing. Sorry, I'm trying to
Yep.
move
Okay.
it, but it's having me draw. I don't know why that's doing that. Where did it go? Because it used to be able to move, but now it's not.
Do you see my?
Oh yeah, now I do. Yeah,
Yep.
I see it now.
So yeah, just so that's what an amide looks like. It's a carbonyl attached to an amine. And then an ester is kind of like a carboxylic acid, except instead of an OH attached to the carbonyl carbon, you have an OR, which means just O connected to more carbons. So, if we go back here, we can see that. Oh, oops. So we can.
Yeah, that's the one I was trying to move.
Oh, yeah.
Yeah.
So okay, so now a lactone is just a cyclic ester. And you can see that this is the ester there, but it's in a ring, so that's now called a lactone. Okay.
Okay, so an ester in a ring is considered a lactone.
Yep.
Okay.
So this is 6-phosphoglucono um lactone. And okay, so that's an oxidation there. Now, next, what we do is, well, in order to do anything about this, we're going to need to bring it back to a straight chain form, which means that we're going to have to break apart the ring, and we can do that with hydrolysis. And so, if you add H2O, the reverse would be dehydration synthesis if it's going backwards. And so um, we're going to be breaking, I'll mark it in this bond over here. So we're going to break that. And so we're going to be left with the that. So you see how it resembles glucose, right?
Yeah.
That we drew all the way here on the left,
Hmm.
except we, instead of having an aldehyde as the first carbon, we have a carboxylic acid. And the last carbon, instead of it just being a hydroxyl, it's a hydroxyl attached to a phosphate.
Hmm.
Now this is just the six. Oops, yeah. So this is now just the six phosphogluconate. So you see it's no longer a lactone because it's no longer a ring, right?
Hmm.
Okay. And now what we can do is we can oxidize this again.
So when you draw that arrow, that means that that's this irreversible.
Correct.
This is, yeah.
Mhm.
Okay.
So we oxidize this again, and what we're gonna do is, um, well, we're going to oxidize. So I mean, okay, there's two things we can oxidize, I suppose. We could oxidize the carboxylic acid group, or we can oxidize the hydroxyl group. Now, the hydroxyl group is easier to oxidize, so that's what we're going to oxidize, and we're going to oxidize that into a ketone. Um, and it's going to be the one, third carbon that we oxidize. So it'll look like this. This one turns into the ketone. And this one, these stay the same. Now, this over here can be the alpha carbon. This one, the beta. And that's why this is called a beta. And then we also have the acid in the first carbon, carboxylic acid. So this is our beta keto acid intermediate. So that's what we have there. And then we decarboxylate because, glucose, we start off with six carbons, and our purpose is to make five carbons. So we're going to have to lose carbon through uh, a decarboxylation. So a CO2 will leave. And the one that will leave will be, which one do you think?
Um, like between like the hydroxyl and the ketone, or can it be any one of these carbons?
It could be a hydroxyl, it could be a ketone, it can be the carboxylic acid. And
Um,
which one, if you look at like the biochem redox that I put all the way on the left, which one, if you oxidize, can turn into CO2?
which to CO2. Um, Oh, it's gonna be, it has to be like the primary hydroxyl that could be eventually oxidized into CO2.
So
So the carboxylic acid.
good, 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 good. So, you know, this one, right?
Yes, that one. Yeah.
Good. So, so now we'll just have the CH2OH, which is just, this one over there. And then our ketone, and then two hydroxyls, and then the CH2OPO4 2 minus again. And now this is, oh, I should ask you for the what? Okay, so the enzymes. So the enzyme for this hydrolysis reaction is called uh, lactonase. And then for this step, from 6-phosphogluconate to the beta keto um, intermediate, uh, what type of enzyme do you think uh, works there?
I'm going to assume it's some type of dehydrogenase.
Yep. So it'll be your 6-phosphogluconate dehydrogenase. Very good. And now for that step between, oh, and then I should say that this is well, I guess before that, um, oh yeah, for the decarboxylation step, um, what type of enzyme do you think that can be?
Like a decarboxylase.
So could be a decarboxylase because that would reduce, I mean, sorry, that would cause a carbon to leave as CO2. But is there any other way of doing that, of generating that CO2?
Um, right now I'm thinking about something back to at the club, like on when pyruvate's being converted to acetyl-CoA, but I'm not sure that's the same type of enzyme.
So remember how I showed you the that like the biochem redox thing?
Um, yeah, but that's what type.
And you were able to determine that the um that the carboxylic acid group, if that gets oxidized, that can
Oh,
leave as CO2.
yes.
Yeah. So you'll see that in a couple of reactions. Another one that comes to mind is pyruvate dehydrogenase complex. So
Yeah.
you'll see in both of these a CO2 leaving, which could lead you to think that it's a decarboxylase, but it could also be um, an oxidation reaction, which would mean a type of, uh, dehydrogenase. So it's going to be still this dehydrogenase. It's just that this beta keto acid part is an intermediate, but it's still like one overall reaction. Um, so it's still going to be the dehydrogenase that causes that.
Yeah, that's what I was like kind of like thinking. I was like, is it the dehydrogenase? But I'm like,
Mhm.
yeah, that was kind of stopping me because I wasn't 100% sure. But
Yep.
yeah.
So, okay. So now this is called our ribulose 5 phosphate. And this can further isomerize to be incorporated in, or rather to can, oops, can turn into the, um, so, so okay, this can then be, reduced back into, hydroxyl in the, uh, last step over here. And so that can be some type of isomerase enzyme to give you your um, ribose 5 phosphate. So that's going to look like this is now ribose 5 phosphate, and this can all get incorporated into like RNA, DNA, ATP, NAD, all of that stuff.
Mhm.
So, um, so, so yeah, that's um ultimately what you will see happen with the pentose phosphate pathway. So you're ending up with the creating a good amount of, oh, I should also say that, um, okay, oops. So the NADPH can function in anabolic processes like cholesterol synthesis, um, fatty acid synthesis, um, reduction of something called glutathione, which just protects the cell from, you know what ROS is?
No,
So it's reactive oxygen species. It's you know what free radicals are?
like a radical, like oxygen radicals with like the don't.
Yeah, like an
Mm,
unpaired electron.
yeah, yes.
So electrons want need to be in pairs. If it's not in a pair, it's like going to be pissed off and just hyper reactive and bad for you. And that's why antioxidants are a thing.
Hmm.
Hmm. So,
And NADPH just like the phosphorylated version of it, or does that P stand for something else? And for an NADPH versus NADH.
oh, um, the yeah. So I can show you.
Mm.
So, so here you can see the comparison of you see what I put in?
Yeah, I see the image.
Yep. So NADH on the left, NADPH on the right, and you could see first of all that they have ribose sugars, right?
Yes.
So that's one of the things that the five carbon like the pentose phosphate makes. And it also makes the NADPH, and you can see that the only difference is it has that phosphate group.
Hmm.
So yeah. So what does that was that your question?
Yes, yes. Um, but I guess like my other question about it is
Mhm.
when would like NADPH be more likely used? Like, why does the extra phosphate make it so that it's preferred to make fatty acids or like um and other things?
Mhm.
If that makes sense.
Yeah, so the fact that it has a phosphate means that you can let's see how I can put this. So these are anabolic reductive processes. So the NADPH would um, reduce any of these, um, and then the NADPH itself would just get oxidized back into NADP. And so essentially what's happening is just that that extra phosphate group makes it so that you can have that reducing power that's going to be like stronger than that of for instance like just regular NAD. Let's see. Okay. So so from what I can see, so it doesn't seem like it's any specific like reason for the phosphate to make a difference. I think it just, from what I see, just helps differentiate it from NADH. So,
Okay.
oh, one sec, let me see. So, yeah, so okay. It seems like the major difference is also is just also so like NADH um functions in catabolism, in ultimately production of ATP. Oh, okay, okay. Because if you think about it, the NADHs, where's the last place that they go?
The electron transport chain.
Exactly. And that's where it gives its electrons to, the complexes, and then those give them ultimately to oxygen, and then, you ultimately produce um ATP that way. Um in yeah, oxidative phosphorylation, whereas NADPH is going to be used in anabolism, and it mostly functions as a reducing agent, um, donating its electrons, and it could just the phosphate can just function as a tag to let it know that it's going to be reserved for biosynthesis. Um, and oh, it can also function as the antioxidant. So those are the main differences between the two.
Okay. Um, well, that makes sense.
Yep. Did you say that you had questions about gluconeogenesis also?
Yes, yeah. So far, like what I got from that is that um apparently it doesn't use hexokinase to dephosphorylate. It uses um
Gluco?
glucohexokinase. No?
Yeah, just glucokinase.
Glucokinase. Okay.
Hmm. So for that, you're going to have um yeah, I can put this. Okay, so this is so you're making glucose from non-carbohydrate precursors like lactate, alanine, glycerol, and these are three carbon compounds.
Hmm.
So, and now there's something called the Cori cycle. So, for instance, um, let's say that we have a muscle cell here. So that's when the glucose through, glycolysis turns into pyruvate. And um yeah, if we're not thinking about like the Krebs cycle, what happens is it'll turn into lactate, and then it'll travel through the hepatic portal vein to this is your liver cell. So you have your lactate, and then it can turn back into pyruvate and back into glucose through gluconeo. I don't know what this is. Consider that. Okay. Neogenesis. And then the glucose could go back to the muscle cell. So this is all blood in between. So it could go back to the muscle cell to break it down and stuff. And yeah, this entire thing is the Cori cycle. Now, if we think about in terms of location where this stuff happens, because that's also important, so we can have the okay, so let's say that we have the mitochondrial matrix, and this will have the malate shuttle and the aspartate shuttle. So we'll have your pyruvate turn into oxaloacetate, which can turn into malate or aspartic acid, which can go, and it just makes those just so it can go through the transporters, the shuttles. So you get your malate and your,
Oh,
yeah.
so malate can, wait, oxaloate can make malate
Hmm.
because at least what I saw in like the Krebs cycle, it seems that malate leads to oxaloacetate. Is it just two different processes that they can go back and forth between each other?
Yeah, so for instance, in the Krebs cycle, the malate can turn into oxaloacetate using malate dehydrogenase, right? Um,
Mhm.
and yeah, it can go in the opposite direction. If you use like the appropriate enzyme. So and even like the pyruvate turning into oxaloacetate, that's going to be a carboxylase that does that this time. But yeah, then they turn back into oxaloacetate, which turns into PEP, and the PEP can turn into uh 2-phosphoglycerate, which can just isomerize into 3-phosphoglycerate. And then this can get phosphorylated again into 1,3-bisphosphoglycerate and into, whoops, G3P, which can, do the thing with DHAP, and ultimately can give you your fructose 1,6-bisphosphate, which can be def dephosphorylated with fructose.
Six phosphate.
So, um,
No?
oh yeah, it'll turn into, yep, fructose 6-phosphate. And this would be a fructose 1,6 bis phosphatase, which means it's hydrolyzing. Um, so adding water and getting an inorganic phosphate out. And then that can enter into your
The
endo.
isomerase? No?
Oh,
Is this
yep.
okay?
Um, but yeah, it'll enter into the endoplasmic reticulum where yeah, it can isomerase into glucose 6-phosphate, and then that's where we have our glucose 6 phosphatase to which is also, a hydrolase that releases your inorganic phosphate to get glucose. And so,
So this is basically the reverse of glycolysis,
correct. And
except for when there's a kinase, there's a phosphatase in its place.
yep. So, yeah. So essentially for the irreversible steps here, we have the, I'll label them in purple. So I think, yeah. So, okay, this one, oh, damn it. So, whoops. So this one and that one and that one. So they're basically the same irreversible steps as glycolysis, just in the other direction. So Oxaloacetate into PEP. This is also going to be, yeah. So the pyruvate carboxylase, actually, like over here, so, okay, pyruvate carboxylates lets CO2 enter or combine with pyruvate. And so this should say is present in liver. So like remember how we talked about like fetal cycle and stuff? And so like so we know in the Krebs cycle that that oxaloacetate can combine with acetyl-CoA to give you your citrate. And
Hmm.
but prior to that, the pyruvate loses a CO2. Uh,
Hmm.
but if we want to go in the opposite direction and go from pyruvate into oxaloacetate, we would need a carboxylase enzyme, and that is present only in the mitochondrial matrix of the liver. So that's why this step would happen in the liver and not, in the muscle cells. Um, and then we also have so yeah, that's what I mean when I say like location is important. And so that's where you can see like what's different with.
Yeah. So, gluconeogenesis will happen in the mitochondria of the liver
So
cell,
yeah,
or okay.
yeah, it'll start off in the mitochondrial matrix of the liver, in order for it to turn into the oxaloacetate, which enables it to turn into malate and aspartate, so it can go through the shuttles, and then it'll turn back into the oxaloacetate. And then when it goes into um, turns into PEP, you're gonna have your um, a well, 4 carbons in oxaloacetate and 3 carbons in PEP. So you have a CO2 leave, um, as like a GTP turns into GDP to give the phosphate to the PEP. So you're
Mhm.
that. And I should have put that for here too. ATP to ADP there. And yep, and then that would give you then that would give you your PEP. And then you look at your between the 3 PG and 1,3 PG, it's gonna be ATP into ADP, because that's where we're getting so a kinase would be doing that. Um, and yeah, and then for this one, it's gonna be an NADH into NAD plus. And yeah, that's pretty much it for that.
Okay. And then it's like one question. So the only like way, because like I see the oxaloacetate, because it leaves the mitochondria, that's why the oxaloacetate will go on to do gluconeogenesis. But if this had stayed inside the mitochondrial matrix, it would have went through the Krebs cycle.
Mhm.
So that's why location is important, because that what dictates what each molecule is going to do.
Yep, and that's how you prevent a feudal cycle. So like so yeah, like in the mitochondrial matrix of regular like muscle cells, which is where cellular respiration mostly happens, you get you don't have the pyruvate carboxylase in the mitochondrial matrix, so the pyruvate just breaks down as you would imagine it to, all the way ending up with the electron transfer chain. But if it if the pyruvate turns into lactate and then travels into the liver and the lactate turns back into pyruvate, then that pyruvate can turn into turn into oxaloacetate with the pyruvate carboxylase in the liver mitochondrial matrix, which lets it then leave and then come out into the cytosol, and the oxaloacetate can then turn into PEP, and then that PEP can turn into like all these other things. Ultimately, uh, you'll see so you'll see like the ones where instead of I think you said instead of the kinases, you just have the um
Phosphatase.
phosphatases.
Okay. That makes that makes sense. A lot of sense now. For
Yep.
why that would be happening. Um,
Here's
s-
like another image here, and you can kind of see the like the different, I guess, directions here. So for instance, the glucose would like you can kind of see like what happens for the actually let me see if I can explain this. Okay, so because it's kind of it's happening like kind of like backwards here, but okay, I'll write this in like blue over here. So first we have, or rather, so this is our mitochondrial matrix, and you have your pyruvate through pyruvate carboxylase turn into oxaloacetate, and they kind of just skip like the steps with like the malate thing, which is fine. So then you see that oxaloacetate can exit the mitochondria, and that's where it can through PEP carboxykinase, which is actually an interesting enzyme because it's both a kinase and a carboxylase because or rather, yeah, a carboxylase. So it can carboxylate things and decarboxylate things, and that's what we have from oxaloacetate to PEP over here. And then the PEP can have go through these reversible reactions that we don't really care that much about, and ultimately go here, and you see a phosphate is removed through the phosphatase, and then this one, the last phosphate is removed through the phosphatase as well. So maybe I'll just put a circle around this to say that this is in the endoplasmic reticulum. Um, oh, and then this actually has in pink the starting material for gluconeogenesis. So for instance, um, we could have over here glycerol that comes from triglycerides, right? That can enter in at this stage where it can turn into DHAP. And then you could also have alanine enter in here to turn into the pyruvate. And then you could also have lactate from that Cori cycle. So what we mostly talked about prior to this was that lactate Cori cycle part.
okay, and so I also saw that other amino acids can enter like the same way that or similar way that alanine does, or is alanine just like the most common, would you say?
Um, so It
Because I
got
remember seeing a list of different amino acids that could be used for gluconeogenesis, but also for another pathway. And there's some that's only ketone, or
yeah.
I don't know if you know what I'm talking about. Yeah.
So yeah, you're talking about glucogenic and ketogenic amino acids. And here, whoops, here's like a list of them.
Hmm. Yeah, I still. Yeah.
So the thing about this is that the glucogenic ones can also just get broken down in Krebs to form the energy. Um, but yeah, typically what you'll see though is because also remember that gluconeogenesis needs three carbon precursors, like molecules with three carbons.
Yeah.
So that also will play a role because like alanine, right,
Hmm.
has three carbons, right?
Yeah.
So like the glucogenic amino acids that are not alanine would have to undergo some more, breakdown and stuff through like deamidation, I think, and stuff like that.
Oh, versus alanine can go straight through. But there are other amino acids that can, but they have to go through another process to get to
Yep.
pyruvate. Or would it create alanine, or would it just go through a different process to get to pyruvate?
Um, so yeah, it can get yeah, deaminated and turned into uh alanine. Yep, it could do that.
Oh,
Hmm.
okay. Um, but it also seems like there's some amino acids that can form acetyl-CoA.
Oh yeah, so that's the thing that everything converges to, like meaning all fats, all carbs, all proteins, the one molecule that they all turn into when you break them down is acetylcholine. Sorry, no, acetyl-CoA. And acetyl-CoA is when, it enters the Krebs cycle and then does its thing and then ultimately electron transport chain. So that's that's what they all converge into, yeah. So like if you're doing like the math for like the total ATP equivalents generated from like a fatty acid, you would like let's say you have like 16 carbons in a fatty acid, you'd do it 8 times or 7 times and you'd be left with, seven 2 carbon molecules that essentially are just a set that you can turn into acetyl-CoA and then put them into the Krebs cycle and then they can make all that energy and stuff. So acetyl-CoA is what everything converges into in catabolism.
Okay, but it seems like for the acetyl-CoA,
Mhm.
it can't go anywhere on this diagram, even though it seems like acetyl-CoA enters into the Krebs cycle though.
Mhm.
So why isn't there not an arrow that will at least point to
Yeah.
oxaloacetate, then move on to citrate. Like, why is that not included on here?
Yeah. So I think they may mean that with this.
Okay.
Here. So like that.
Okay. I was about to say, I thought that was a thing. And then, but I see on their Krebs cycle that they skip the succinate. Like after the succinyl CoA,
Yep.
doesn't it go to succinate,
Yeah.
then fumarate?
Yeah.
So is the succinate just not really important? Is it more like an intermediate that's not
Oh no, this is just like a condensed Krebs cycle just to show you like the glucogenic and ketogenic amino acids. So it's not the whole Krebs cycle. It's just like a shortened version of it.
okay, okay. That makes sense.
Yeah.
But okay.
So, like it could be more so like something.
But then again, when, like for example, when would arginine be needed to create alpha ketoglutarate if we already have citrate? Is it only if citrate's not there, you can use one of these amino acids to keep going? Or what's the point of having arginine or glutamate or something like that?
Oh yeah. So yeah, those would go through like various um like transaminase or deaminase kind of reactions. So um which would be like so let's see if I can find maybe example of that. So like
And also, like, can you send me this like whiteboard by chance? Or
at the end, like I just have my students export it into PDF. So
yeah, okay.
okay, so this is actually so um the urea cycle. So that's when you have your protein, it's a catabolism of protein. So you hydrolyze your proteins into your amino acids.
Mhm.
And I mean, these can also be going towards protein synthesis. Oops. But the amino acids, I'll do like a condensed one here. So like there's phase one where they turn into alpha keto acids. And then there's a phase two in which they'll turn into Krebs cycle intermediates. And then a phase three where they ultimately, end up as ATP and CO2 and water.
Yeah.
So, and now an example of this could be like, be this.
I see it. Yeah.
Mhm. So so here you can see specifically that like alanine, glycine, threonine, cysteine, serine, tryptophan can turn into through the urea cycle, turn into pyruvate. Uh,
Mm-hmm.
how like tyrosine, phenylalanine would turn into fumarate. So
Mm-hmm.
they're going to turn into various intermediates, right? And that's how they can enter in as the various intermediates after they get broken down.
But then I guess like my question is, why would they need to do that? Like,
Uh-huh. So,
like for is it because if there's no isocitrate or citrate because that
so
will allow, sorry, yeah,
yeah.
keep going. Sorry.
Yeah, so ult the catabolism ultimately is going to be involving the Krebs cycle and an electron transport chain. So so I mean, we typically think of just like glucose that goes into the Krebs cycle and electron transport chain and gives us energy, but really everything does. So if it's a longer sugar, then that can work too. If it's amino acids, those can also, and same thing with fats. Like they'll break apart into things just so they can enter into the Krebs cycle, just so they can go through the electron transport chain. Because cellular respiration, that's the overall thing that happens in a cell, and that's with some type of hydrocarbon plus um oxygen yield CO2 and water. So that's cell respiration. And so that hydrocarbon, which we typically think of as like glucose, that could just be malate or fumarate or oxaloacetate or alpha ketoglutarate. Those are the things. So like ultimately everything everything catabolic will have to still undergo the Krebs cycle and electron transport chain to give you your energy.
But then where are the electron carriers coming from? If let's, for example, like with the amino acids like entering in as alpha ketoglutarate, it's going to have less NADHs. So then where is it coming from? Because like when we go from isocitrate to alpha ketoglutarate, that's where we have an electron carrier. So
Mhm.
we're short two or so.
Oh,
Um,
they're just not written here, but basically you're doing something that will turn that glutamate into the alpha ketoglutarate, and the alpha ketoglutarate can it's not written here, but it'll just function like it would in the regular Krebs cycle, meaning the NADH and all those things. Because the NADHs and all those things, like those are things that happen in your so here, this could be an example. Let me see.
so it's still going to create electron carriers, but like
Mhm. Yep.
okay. So,
So,
but would it create like the same amount of like if we were to go through the regular one with glucose that we know?
mhm,
It would still create about like two NADHs, the
mhm,
CO2 as well.
mhm.
So we would still have the same ending products,
Yeah.
just different starting.
Mhm. Yeah. So I mean like, for like it'll just match like the ones that have the same number of carbons. So they can enter in at different stages of that as intermediates. But yeah, ultimately you're going to be still producing the NADHs and, you know, FADH2s and things like that. So, because, yeah, because what you're getting rid of in the urea cycle is well, the urea, right? So and ammonia. So that's what you'll see. So another example of this could be like, let's see. So, okay, when the amino group is removed from an amino acid, it's converted into ammonia through the urea cycle. The remaining atoms of the amino acid result in a keto acid, which is a carbon chain with one ketone and one carboxylic acid group. Um, the liver can synthesize urea from taking two ammonia molecules and a carbon dioxide molecule. And so urea is that primary waste product. And the amino acids can enter in as pyruvate or acetyl-CoA or all of those, you know, intermediates. And so an example could be asparagine that when it's deaminated can be converted into the oxaloacetate or the aspartate because those have, I believe, four carbons as their chain, right? Yeah.
Oh, I see it. I do it here.
Mhm. So we see the glutamate into the alpha keto glutarate
Yeah.
and 1, 2, 3, 4, 5 glutamate, 1, 2, 3, 4, 5 alpha keto glutarate for the because yeah, if you remember like this was like 4 carbons, 6 carbons, I think five or no, that was just the isocitrate. Still 6 carbons still, and then 5 carbons. Then I think this is the 4 carbon one. 4 carbons, 4 carbons, 4 carbons. So yeah, so if you're so you get rid of the like you're just left with that like side chain when you get rid of the um amine or you get rid of the amine group and then yeah, just a matter of however many carbons the molecule has. So like glutamate has, these 5 carbons, so that's how why it would enter into alpha ketoglutarate, whereas serine has 3 carbons, and that can enter in pyruvate, which is 3 carbons. Um, and then you have and then just like and then amino acid oxidase is when you turn an amino acid into the alpha keto acid. So like a good example of that is like alanine, because you can see that here. So for us, we're going to go to the left. So from alanine, if we go to the left, we get rid of the amine group and we are adding or replacing it with a like a ketone, and that gets you pyruvate. And so that's what's meant by the where they wrote somewhere the alpha keto acids. So this is the was that me? Was that you?
Huh?
Oh, I think that was me. Okay.
Yeah, like I didn't touch anything, so yeah.
Yeah, yeah. So sorry. So this is our amino acid, and then this is our alpha or what was it? Alpha keto acid. So yep.
Mhm.
So yeah, that's how, and this probably could also help here. So this one you could see alpha amino acid through transamination can turn into the alpha keto acid, and then you can have uh it turn into like let's say glutamate um if you go in the opposite direction. But yeah, ultimately you're going to be getting rid of in an amino acid, you're going to get rid of the amine group as ammonia, which and you're going to get rid of the carboxylic acid group of the amino acid, um, which will be the CO2, and those can combine to form the urea. And so the side chain is ultimately what you're left with, and that can enter in at various stages of the Krebs cycle.
Mhm. Okay.
That kind of answer your question?
Yes. Yeah.
Mhm. Good. Okay. So, yeah, how do you feel about these?
Yeah. I feel like a little better about it, especially stronger, specifically with like the Krebs cycle. and gluconeogenesis and how that relates to glycolysis.
Hmm.
I think it's just more or less, especially for the pentose phosphate pathway, more
Hmm.
like repetition really. And there's trying to get those down.
Hmm.
Um, and then I know like well, the rest when it comes to like biology, it's more or less the systems, which I think I could get that on my own. Um,
Yeah,
but this got through like a bunch of the pathways of where I was struggling with. I
yeah.
think the last one that I needed, I'm gonna try to go over myself is the fatty acid
Mhm.
um synthesis.
Mhm.
And I think that's the last metabolic pathway that I need. I
So
know it's
yeah, so there's going to be the that beta
yeah,
oxidation,
beta oxidation. Yeah.
um, and then I would just say like to have like you don't need to know it at like as much, but like just the basics of like uh glycogenesis, like the making of glycogen, which is just glucose, polymerization. Um, Glycogenolysis, which is like the opposite when you're breaking down glycogen into glucose monomers. Um, and yeah, I think that's the only other one that I could think of right now.
So I got through at least the bulk of it, but I still kind of have a little bit more to go, but at least got through
Hmm.
the bulk of the metabolic pathways. So it's not too bad. I think it's more or less repetition.
Yep. Yeah. So I would say to know the what you need to know the most is going to be your glycolysis, your
Yeah.
Krebs cycle, your electron transport chain, your gluconeogenesis, including the Cori cycle. Um,
Hmm.
and then like second tier would be pentose phosphate pathway beta oxidation. Um, and then third tier would be like, or maybe in the second tier, I could also add in like urea cycle. And then third tier would be like the glycogenesis, glycogenolysis kind of thing.
Oh, so glycogenesis was like a third tier. Okay. Well, but still, they're still important, but
Hmm.
I can see, like, let's see why. So I can make this into a PDF. How do I go about doing that?
Yep. So on the top left, there's I wrote metabolism 2 and there's three horizontal lines.
Yes.
Click that drop down menu, and then it'll say export, and you can export it into like a PDF.
Drop down menu.
Mhm. Like if you click the three horizontal lines,
I click the three horizontal lines. I see whiteboard.
do you see export?
I only see whiteboard history, but I do not see an export on here. Like
Okay.
I see a view. Mm.
Here, I can.
I see a saved thing, but like, hmm.
Here, I'll export it myself, and then I will put it in the chat.
Okay. And then like, how much for this session?
Um, yeah, one sec. You see. what I sent in the chat?
I am saving it
Mhm.
to my down. Yeah, and I have it saved.
Okay, and then let's see. Should be 126 for today.
Okay. It's at 126. Did you receive it?
Yep.
Okay.
Awesome. So I will see you on Saturday at 8 p.m. Eastern.
Okay.
All right. Have a good one.
You too.